categories, so I'm familiar with the tools, but I wouldn't say that I am a category theorist. And I'm not a neuroscientist, but I've been working for the last 3 years and a bit with the people at the Blue Brain Project at UCSL on projects related to neuroscience and using topology to understand neuroscience, or at least a little bit of neuroscience. Working also with other neuroscientists who actually have wet labs and things like that. So there are actually a number of different projects we have applying algebraic topology in neuroscience. The few results of ours that I'll be talking about towards the end of this talk are coming from the collaboration with the Blue Brain Project. So just a few words about what the Blue Brain Project is. It's a project funded by the Swiss government that's been running for about 12 years now, something like that, that is working on reconstructing, doing a digital reconstruction first of the brain of a rat, taking into account as much biological data as possible in this reconstruction. And then running simulations and such on this. So, but I'll talk about more in detail about that towards the end of my talk. So what I'm going to start by is giving an introduction to some basic neuroscience. Again, with the caveat that I'm not a neuroscientist, I'm going to tell you things about what I've learned over the last several years, or 2 years. And then I'm going to talk about some work that's already been done on applying categorical to neuroscience, some first attempts, which have mostly been on the levels of cognitive neuroscience rather than the level of neurons and so on. Then a bit of sheer speculation about what category theoretic tools might prove to be useful, what frameworks might turn out to be helpful within the context of neuroscience. And then at the end, I'll explain some of what we've seen using the tools of algebraic topology in neuroscience, which may be a source of inspiration for some of the things we can do in category theory. So, neuroscience with a working category theorist. So I'm not assuming that you know anything about neuroscience except the fact you have a brain. And so what we're going to do here is I'm going to tell you something about the basic components of the brain. The neurons, the nerve cells, and their components, what they're made up of, how they work, how they transmit signals around the brain. We're going to completely ignore a good deal of what's in the brain, which is called the glia and vascularization, so how the blood supply gets to the brain, which is an extremely important part of it. But this, just taking into account neurons and their neurites and all of this and how that works is complicated enough without going into too much more detail. So, let's talk about neurons. So one great advantage of working people at the Blue Brain Project is that I work with some fantastic visualization people. So they've done this amazing, the previous 2 images as well, but this is a visualization of 2 different neurons who are talking to each other. So neurons have sort of 3 basic subparts in the structure. There's the cell body, which is called the soma, and they have, so here we have the somas. And they have sort of incoming wires, which are the dendrites, and outgoing wires, which are the axons. And along these axons and dendrites, there are connection points. You see some that are sort of lit up here, which are called the synapses. This is where the axon of one neuron meets the dendrite of the next neuron in the sequence. So they actually have these sort of directed connections, between these neurons. So if we wanted to represent this, sorry, I'm going to have to work on the board now. So, you can think of, here's my cell body, so the soma, S, and into the soma they're feeding a bunch of dendrites, like this. So these are my outcoming guys. Incoming guys, rather, and then I have some outgoing guys, which are the axons. Like this. And on the dendrites and on the axons, you have these connection points, which are called synapses. Like this. And also on the axons here, and these would be the dendrites of the next neuron sequence.
Many synapses each?
Yes. They have, in general, thousands. So to have some idea of the orders of magnitude we're talking about, in the human brain, so the neocortex, the part of your brain that's involved with higher cognitive function, we're on the order of hundreds of billions of neurons. And these neurons have hundreds of trillions of synapses. somewhere on the order of 1,000 synapses per neuron in this case. And when you have two neurons that are connecting together, so the axon of one and the next, in order to form a reliable connection between the two neurons, you need actually to have several pairs of synapses that meet. If you have just one synapse on the axon of one, and one synapse on the dendrite of the other, that connection is not going to be reliable. So you're not going to be able to say, okay, these two guys actually communicate. To have a reliable communication, you need something on the order of 6 or 7 different neurons that are synapses. And so one way of representing this kind of structure is to say, well, let's look at it in just the most simplistic way. Think about it as a directed graph, where the vertices of the graph are going to be the neurons, and the edges, the directed edges in the graph, are going by connections between neurons. So that's a bunch of synapses that are creating a connection between two neurons. That's sort of the coarsest grain level, which we're going to look at in a network of neurons like this. And as I'll explain in a little while, there are good reasons for looking at it a little less, a little more fine connection. But that's one way to think of it. So one could represent it like this, where you have a neuron here, and a neuron here, and a connection between the two of them is made up of a certain number of synapses. And so what they usually say is the one before the connection is called a presynaptic neuron, and the one after is called postsynaptic. So one thing to remember, so I said that if we want the connection between the two neurons to be reliable, then we need to actually have several synapses that are used to communicate between the two neurons. This is because the behavior of these neurons is very stochastic, highly stochastic. You can even have, you can be sending out a very strong signal from the presynaptic neuron, and the postsynaptic neuron will say, I don't care, I'm not going to fire. So you have to take into account the stochastic element in these connections. They're not very reliable. Okay?
Sure. So if the neuron receives a signal through one of its dendrites, it will then do something and emit a signal on its axons. Does it make a difference which dendrite the signal is coming in from?
Yes. Yes. We're going to go into some more detail about this. This is something that's really interesting, actually. Let's see, what else I want to talk about? So let me say some word about how these neurons are communicating between each other. So they're communicating electrically, so chemical signals, actually, ions that have been transmitted through ion channels in the synapses. And so what happens is you have to reach a sort of a threshold level of potential here in this neuron, and it'll do what's called firing and action potential. It'll send out an electrical signal through the axons, and that's called spiking. So they communicate via this kind of spiking. So I'm also neglecting something that's called a gap junction, where you have an electrical connection, a direct electrical connection between the neurons. We're focusing purely on these chemical synapses. But there are also electrical synapses. Okay, it's already complex enough like this, I guess. So, as I said, we're going to focus on cortex, which has roughly 100 billion neurons in humans. So the cortex is formed of actually 6 layers. Outside to inside, it's this thin gray this thin layer of gray matter you have on the inside of your brain. So it's 6 different layers, and the different layers have different functions, and they're composed of different kinds of neurons, which have different kinds of structures, different shapes, and different electrical activity. So what this is, we can talk about the different morphological types of neurons. So here we see the 6 different layers. 1, 2, and 3 are often grouped together. 4, 5, and 6. So this is from the outside to the inside. Looking at these layers. And what we have here is, first, there's a separation into what are called excitatory and inhibitory neurons. So even though there's a sort of less variety among the excitatory neurons, they make up about 80% of the neurons in the cortex, and the inhibitory is about 20%. And so what we see here is the morphological diversity of these neurons. So I do this very simplistic picture, but we see that actually here, the axon actually has a quite a complex structure, as does the dendrite. The size of the neurons changes quite a lot, and so on. So as you can probably tell from their names, the excitatory and inhibitory signal all along, and the inhibitory ones are like the parents of a teenager who say, turn down the music, please. Don't let things out of control. So you really need a nice balance between the excitatory and the inhibitory neurons in order to get good behavior and result. Yes?
All right, you can throw it for a second. Did anyone park here?
Any question?
It looks like that with the excitatory ones, it looks like the dendrite sort of only extends in the top half of it. And I'm wondering if there's some, but yeah, there's something around that.
Well, okay, so you do have some that are some extending downwards. And there's another one that's actually not listed here because it's sort of more, this is a figure from a paper in 2015, and there have been sort of more in-depth analysis of it than then. There are also ones that are called HPC neurons here, horizontal, they're just saying that dendrites are horizontal. So, but it's a good observation that the dendrites are often going upwards to these higher layers in order to show
What's also plane of this image? This is a three-dimensional.
right, so this is like you're taking a slice like this.
Which way is it coming?
Okay, so I'm terrible drawing, so I'm not going to tell you draw. So here you have your brain, and it's like you're doing a vertical slice in your brain. So this is like the outside to the inside. Okay. And so this really is sort of up. Any further questions? So this is just to give you some idea of how diverse the neurons are in the brain, just in terms of their shape. So we have 55 different types here. And I hope have time to explain at the end of the talk, this morphological diversity is actually very, very important for ensuring that you have sufficiently complex structure in the brain.
Sorry, I'm very confused about this. I would have thought these connections between the neurons, with respect to where the axons are going, the dendrites are going, would be very, very complex and interwoven. But all of these have got a very neat directionality with all the axons on one side of the dendrite.
Well, okay, so this is
some artificial.
This is a somewhat artificial representation, and this is, I suppose that in fact they in three dimensions. Here we're just looking at a real slice, but there's a fairly neat our orientation preference in your geometry.
The right left, though, I think, is just so you can see the difference. Yes?
Yeah, basically.
So the front orientation is top down.
I'm sorry.
That's the top of your brain. Further in.
Sorry about
Okay. All right, so that's those morphological types. Then life gets even more complicated because there's also diversity in the electrical behavior of the neurons. Because the way they're communicating is by transmitting electrical signals, so their actual electrical behavior is important. So what you see here is the possible ways that a neuron can react to a certain incoming current. So they do these experiments where they have, put in a little glass electrode into the soma, and you just feed it with a current, and you see how does it react. And so it has these different kinds of patterns of electrical reactions to this incoming current. And the point of the second panel of this figure is to say, okay, even if you fixed one particular morphological type, in this case it's some sort of basket cell from layers 2 and 3, then that particular morphology can have different kinds of electrical behaviors. And so they can have, once you take into account the diversity of the morphologies together with the diversity of electrical types, suddenly you have an incredible diversity of possible neurons, how they can fit together and what their electrical behavior is. Okay. Now we want to talk about plasticity. So, one of the important things about your brain is that you can learn, presumably. If we're all here, we've all learned various things, at least about category theory. So there's a lot of discussion in the neuroscience community about what learning is, how to capture it, what it really means to learn. And one can argue that the underlying biophysical mechanism of learning is what's called plasticity. So the synapses have various types as well. There's a great diversity of synapses. And one thing that one needs to take into account with the synapses is how strong they are. That is, how reliable they are at firing, and so on. There are various measures of the strength of a synapse. And also, in some sense, its strength depends on, in terms of how it's going to amplify signals, how far it is from the soma, its actual path distance along the axons and dendrites. And so one thing that happens when the brain is exposed to repeated stimuli is that synapses can become stronger or weaker. And that somehow if you want a connection to become more reliable, you want the synapses that are forming that connection to become stronger. So if you have, and so the scientists at the Blue Brain Project have done an experiment, a simulation they did on one of the big supercomputers at Argonne National Laboratories, where they presented their digital reconstruction of the circuit with a sequence of repeated stimuli. So it's like you're listening to a sequence of 8 tones over and over and over again. What happens after a while is that certain synapses stop reacting to some of the tones and react only to one of them or only to 2 of them. So they become preferential for various tones, and they become stronger or weaker in response to that. And so what we see here is, this is just an example of the sort of neuron, sort of pyramidal cell they were looking at. And with this release probability is some sort of way of measuring the strength of the synapse. And this is time going along here. So they're repeating this stimulus over and over and over again, these 8 tones over and over, and seeing what happens to the strength of the synapses. So at the very beginning, what they did is just sort of a random Gaussian distribution of the strengths of the synapses, and then said, okay, let's go. Let's just listen to these 8 tones over and over again. Oh, sorry, I take it back. This one is spontaneous activity. So they were not listening to tones. They were just let the brain do its stuff. Just think, just sort of spikes being fired randomly and so on, and see what happens. And what they found at the end of the simulation was, even if they had to sort of give a nice so kind of Gaussian distribution of the weights at the beginning, they tended to form this sort of bimodal distribution at the end. That they just sort of naturally separate like this into weak and strong. On the other hand, so this was spontaneous, if you actually do this sequence of 8 tones and you see what happens after you've listened to these 8 tones over and over again, then you have a very different kind of distribution that happens. You get this weak and the strong, and then also this sort of thing in the middle. And so just to show that there is something different that's going on in the way the synapses are reacting to a sequence of tones that it's learning, so to speak, compared to when it's just sort of not reactive, no particular incoming stimuli. So that's one experiment that was done, and we're trying to learn more about exactly what's going on in plasticity, what it means that there's this new kind of distribution, and we're looking at this with tools from topology right now. But that's an important part of the learning process. Yeah?
Sure, this was all digitally done?
Yeah, this is all digitally done. So there are experiments that have been done, wet lab that have experiments that have been done to say, you take actual cells in vitro, and you repeat some particular, and you see what the results of those wet lab experiments were built into how this digital simulation was done. So whether this is exactly or really what's happening in biology, it's just like the best approximation you can do right now. Yeah?
Can you play with neurons in vitro, or you have to use animals for that?
You can play with neurons in vitro, yes. So your experiments in a room where it's 38 degrees Celsius, but it's but you can actually, yeah. Mm-hmm. Any further questions? Yeah. About the actual model, I can say more about how the plasticity run was done. Okay. Other questions?
So you seem to suggest that there is a connection between the strength of this connection and learning.
Yes.
So learning is increasing the strength, and when the connection becomes weaker, that corresponds to forgetting it?
Not necessarily. I mean, it could be there's certain patterns, firing patterns that you want to strengthen. I mean, the connection between two different neurons, but often in order to learn something or to remember something, you need to have a sort of particular pattern of neurons that fires, they call these engrams, that fires more often. And so you want to strengthen particular firing patterns. And that may mean that you want to weaken other things. So there's a saying in neuroscience, let's see, the two neurons that are firing quick, then the connection is going to become stronger. Okay?
Also can mean many things.
Yes.
Just remember what is it closer to?
So I'm not enough of an expert in learning to so this is, I mean, I agree completely that learning is a word that has many possible meanings, and this is just one, I would say, is like a biophysical foundation for higher notions of learning. Yeah. Okay. So another thing that you observe when you look at this plasticity simulation is that at the end of the simulation, so it's been running for 1,000 seconds, the strength of the synapses varies a lot by position on the axons and dendrites. So we have these sort of average ones that are down here, and then out here you have sort of a mix of stronger and weaker ones. And so this is another indication that on a single neuron you can have very different kinds of behaviors of the synapses. Some can be a lot stronger, a lot weaker. And this is related to, so this what I've been talking about for the last few slides is work from the recent PhD thesis of Giuseppe Kindemi, who's one of the PhD students at the Blue Brain Project. And in another thesis project by Willem Widbo, he was looking at what the dendrites are actually doing. So it's a relatively recent discovery that the dendrites are not just acting as wires going directly to the soma, just sort of blindly transmitting signals. There's actually computation that's going on in the dendrites. So there are groups of dendrites that are working together as processing units and that are taking information in. So one thing that Willem was studying this idea of dendritic computation in his thesis, and one thing he looked at was what's happening in plasticity, and this was an example. So this is again a simulated exercise. Again, the same kind of pyramidal cell, and he was saying, well, you know, at the beginning, if you can have this sort of mix of different kinds of synapses here. These guys are kind of acting as a shunt, and there's a kind of a biological backprop that goes on during this process, so that at the end, the synapses kind of sort themselves out. So you have on one branch they're doing one kind of thing, and another branch they're doing another kind of thing. And so this is, he calls this branch-specific learning. So there are different parts of the dendrites that are learning differently. And the way it studies this is by looking at this arborization. So these dendrites really form a kind of a tree. Or you can say the root is the soma, or the cell body. And from that, you can sort of take this tree, something like this, and with its synapses located in various places. And what he considers is a kind of cover of this tree. So he can say, well, maybe I have a piece like this, and I have a piece like this. And he's looking at what's going on with the behavior. He's doing what he calls these dendritic compartments. And what it looks like, and he didn't even realize this. I was on his thesis, he was looking at this thesis, and I said, oh my goodness, because what he was actually looking at in the end to describe the kind of rules that this compartmentalization had to follow was exactly a co-cycle condition for this. So I think that, and this is not something that anybody has explored or thought about in any detail, but I think in order to understand this dendritic compartmentalization of computation, one way to approach it is going to be through sort of a sheaf theory or a bundle theory or something like that, in order to understand what's going on here. Most of the neuroscientists, no neuroscientist I know has these tools in order to do this. But he wrote down the co-cycle condition without knowing what he'd written down. Sorry?
An n co-cycle for which value of n?
2. Yeah. So there we go. I think they're really interesting things to do here, and it would be great if somebody did some thinking about it. But anyway, we have this notion of important compartments within the dendrite to think about. Which means that if we really want to think in detail, let's see, where's the eraser? Yes. If we really want to represent what's going on with these neurons, the picture is going to look more like this. So here's my cell body, my soma, and then feeding into this are going to be dendrites, yes, but it's going to be more like this. I'll have like some sort of dendritic compartment like this, but there can be also previous dendritic compartments that are feeding into this one. So if you really want to get into detail, you're going to have things like this, and they haven't even really started exploring what happens on this end yet. Whether there's also compartmentalization that goes in the axons, I don't know. I don't know if anybody knows. But they do know that complicated things are going on in the dendrites, like this. So, if I can wake up my computer. Okay. So, there's a lot of interesting between them. And if you really want to get serious about it, then you have to start saying, okay, but there are also weights we should be putting on here to take into account the weights of the synapses and such. Yes?
That again comes from a simulation, or is
Right, this is again from a simulation. It's again taking into account studies that have been done of, so people have actually observed this sort of compartmentalization in wet lab experiments, looking at things in vitro, and then saying, okay, let's take as much as we can. They're using some sort of cable equation to analyze this, and so it's a combination of the mathematics that goes into it is very much numerical analysis and so on, but it's coming from actual wet lab observations.
Because if it's a simulation, it raises the problem of what is the initial state? Because here it seems to be indicating that before learning there was some state, and after learning there's some state. It's hard to imagine a neuron that has never learned anything.
Indeed, absolutely. And so that's why in the reconstruction that I'll talk about at the end, one thing they did was they were specifically modeling the brain of a 14-day-old rat, which is before the rat has ever opened its eyes. So at least has had no visual stimuli and very little other sorts of stimuli. But yeah, the zero state doesn't exist. So you just have to choose some state and then see what happens. Yeah?
Curious, thinking about biological background, I might be too aside from the talk, but one clarification, is it that through learning that's how you get the dendritic compartments?
No, so the dendritic compartments are already there.
Okay.
And then it's just that through plasticity, the behavior of the synapses within each compartment can change. Yeah?
I'm not sure if I understood it properly. You're saying that in the dendrite there is some sort of computation also being performed.
Yes.
And also in the cell body, which is...
Yeah. The cell body is sort of where things converge.
Can we abstract that? It's all just with the egg of some sort?
Sure. I mean, you can,
Or is it fundamentally different things happening in the dendrites and the cell?
fundamentally different things that are happening in the different sorts of computations that go on. So I think, so again, this is going outside of my comfort zone, but I think that they have a pretty good idea what sort of computations go on here. And they're just starting to think about what sort of computations go on here, and it seems to be of a different nature. But I know I'm being filmed, but I wouldn't quote me on that. But there's still like very, very much they don't know about what's going on here. It's only a relatively recent discovery that there's any sort of computation going on here at all. Okay. Further questions? Okay. So that was all on the level of neurons, but of course what's easier for people to observe, other than when you're looking at individual rat neurons under a microscope or something, is to think about actual brain regions. So there's a lot of research that's been done about thinking about connections between brain regions. So I stole this slide from a talk by Olaf Sporns at EPFL 2 years ago. So this is another Lausanne colleague, actually, Patrick Heimann, who is really a world expert in what they call connectome. So Patrick and Olaf invented the word connectome simultaneously. There were two papers published in 2005 where they both used the word connectome, and it's like, okay, great. So I guess it's a concept. And so what we're seeing here is how you can come up with some sort of structural connector for the whole brain in terms of its regions. So it's a relatively complex process involving MRI and doing diffusion spectrum imaging and tractography and so on to come up with some sort of partition of the brain into, here's roughly 1,000 what they call ROIs, regions of interest, and they figure out the connections between them using this tractography. So that's looking at the white matter fibers and how water is flowing through the white matter fibers, you see the connections between them. This gives us a graph, but this graph is now not a directed graph because they don't really know which way the information is flowing between two regions, but they know that the regions are at least connected. And so this is there's a lot of research that's gone into thinking about these sorts of the connectome at this level. And what one is really interested in is, structure and function, right? So on the level of neurons, we can say, well, we've got the neurons that are connected together and their signals firing around, and you'd like to also understand, what the relationship is between this structural connectome and what they call the functional connectome. So when you're doing MRI, they look at what they call the BOLD signal, the blood oxygen level something, and that then tells you how actually activity is moving around the brain, and they look at like a Pearson correlation or something like that between the BOLD level in various parts of the brain, and then they come up with, again, an undirected graph describing connections between these areas that has to do with the function, has to do with the activity. And then they want to compare the structural brain network from the previous slide to the functional brain network, which is coming from this time series data of these BOLD signals in the brain. And you want to say, okay, well, can I deduce something about the functional network from the structural and vice versa? So this is kind of one of the holy grails of neuroscience, to understand how structure shapes function, and then when you start thinking about plasticity, how function then modifies the underlying structure. Okay. That's what I want to say. Another important thing to know about the brain is that it's highly hierarchical. So if you start looking at different regions of the brain, there can be important subregions. They're very natural ways of dividing the brain up into different modules using different modularity criteria. And here, for example, they have one, if you're looking at MRI data, so what you've got here is some sort of heat map showing the correlation between the different regions. And then using this, in some particular modularity criterion, you can see that this graph divides up very nicely into these different modules and so on. This is looking at this diffusion spectrum imaging, which is another way of looking at the connections. So you have this hierarchical structure, and this is just an example showing these different regions of the brain and how they form different modules. This is in a young child, and this is in an adult, showing that the regions, actually, the modular structure changes as we get older, which is probably not surprising. But there's this notion of hierarchical structure, which is extremely important on this level. And we can see the same kind of thing if we start bottom up with the neurons, that they naturally sort of cluster into different groups. And you can say, well, you could imagine starting with a network of neurons at the bottom and start clustering, and then imagine, see whether you could end up with this kind of modular structure at the top. So this is another example showing this hierarchical structure, showing the connections between all the cortical areas and how some of the cortical areas then divide up into different modules like this. And this turns out to be very useful for thinking about things functionally. So this is something that should be taken into account, I think, if one really wants to get a whole brain kind of picture of what's going on. About really a hierarchical network. So, I want to talk to you briefly about some of the approaches that people have tried using category theory in neuroscience so far. So far, the way I looked at this was to actually do a Google search, category theory in neuroscience. There are surprisingly few hits, actually. Be really great. 10 years from now, there were a lot more. But a lot of them have been sort of very descriptive of people say, well, it should work like this. Here is a framework that would, that the brain should fit into. And in general, offering very few tests to see whether the framework actually makes sense. So it really kind of depends on what your goal is. I mean, having discussion with David about this the other day, should the goal of applying category theory to neuroscience be about finding a way of describing an actual functioning brain of a mammal or whatever, or a fly, Or should it be, I want to try to construct a network that can learn or can think and whatever, in which case finding something that's, this looks like the way one could describe the brain, maybe it's good enough. So, one of these is what's called memory evaluative neural systems. So this is due to Erisman and Van Bermersch, I guess. And so if you look at the papers, so they have a whole book on memory evaluative systems without the word neural. And an evaluative system, it's, as far as I can tell, I admit I didn't spend that much time reading about it, just seems to be a functor from the poset of the real numbers to a category of small categories of what are called partial functors. So these are functors that define not necessarily on all the objects, but on some subset of the objects of the source category. And what they claim is that you can come up with a formal unified model for studying the brain if you use these evaluative systems. And they have what they call the complexification process, which is going back to this idea of hierarchy. They're saying, well, when you want to form important areas of the brain, then what you're going to do is you're going to take colimits of various pieces, and then you take colimits of those colimits, and you take colimits of colimits of colimits, and so on. And this is creating increasingly complex subunits that are all working together, and the connections between them are described by this, the graph. So, and they have some idea that this should lead us to a way of describing what these higher cognitive processes, so thinking and speech and all the sorts of things, from this sort of complexification process. So this work has been cited a fair amount, but again, one thing that I've learned to appreciate since getting involved with neuroscientists is that it's very easy. I mean, I was playing games with thinking about what sort of topology might be involved in neuroscience for a number of years, to just, create beautiful theories like this that have zero to do with reality. So,
which year was this?
So this was published, this was in the early 2000s, I think. But Erisman's been thinking about this for a while. So the book about memory evaluative systems is sometime in the late 90s or early 2000s. Then there was an article in PLOS Computational Biology, which is probably the only time an article in category theory has been published in PLOS Computational Biology, as far as I know. It's where the authors were considering the notion of systematicity. So this is where I'm really very far out of my comfort zone. I am very much not a cognitive neuroscientist, so I'll do my best here. So systematicity is about clustering of cognitive abilities into related behaviors. So the example that they give in the paper is to say, suppose you understand a notion like the class of people, and you understand a notion like A loves B, to mean that A is the lover and B is the loved one. That means then you can take sort of any A and B from your class of people and put them in the slots for A and B, and you can deduce what they are. The important thing is you have an ordered pair of elements. And you know that the first one is going to be the lover and the second one is the beloved. And they say that, well, this notion of imposing an order on it is sort of arbitrary. Where is this coming from? Well, we should think about that in terms of the diagonal functor on the category and the product functor. So we have an adjunction between the diagonal and the product, and that's what's underlying that kind of systematicity. And then point out that cognitive abilities are often not truly systematic in that sense, that there can be additional criterion. So the other example that I give is thinking about the notion of offspring. So you can say that a colt is the offspring of a stallion, or a filly is the offspring of a stallion, but it's certainly not going to be a heifer is the offspring of a stallion or something like this. You have to take into account as well the particular species you're talking about, so that the kind of construction that describes that kind of quasi-systematicity is more of a pullback over species of the whatever notion you're talking about. And so they're saying, well, okay, we again have this sort of universal construction from category theory, a pullback that's describing the framework for quasi-systematicity. What their conjecture is that these systematic and quasi-systematic properties of human cognition are examples of universal properties of different sorts, often characterized by adjunctions. And then they conjecture as well that learning and development in a human is a process of acquiring universal cognitive constructions like this, and in particular, adjoint cognitive processes. So I think it's an amusing read. It's a nice read, and who knows? So when I was preparing this talk, I ran these articles past a colleague of mine who is an eminent neuroscientist, and he said, well, maybe. But he's willing to sit down and talk with me about it and see whether category theory might actually be useful to him. I mean, he's very mathematically open-minded and said, well, why not? But he said, be careful, cognitive neuroscience is very, very, there's a lot of theories, and not all of which can be tested very easily. Another notion, which is again rather controversial, is what's called integrated information theory. So this really, oh boy. Now we're getting to philosophy. So we talk about the notion of, I guess it becomes quale, a single moment of whole experience. Okay, so we want to understand consciousness in terms of qualia. That's the point of this integrated information theory. So the output of this sort of theory is what they call a maximally irreducible conceptual structure. So they have some sort of state space, which is talking about, the temperature and the color and the day of the year and all, whatever that is. And so you have a constellation of concepts within the state space. And they say that these mix are identical in some sense, which is sort of a mysterious, what it means to be identical, to qualia. Okay. And then they have 5 essential components of consciousness: intrinsic experience, this is your own personal experience; compositionality, you can put different pieces, different sort of qualia in a narrow sense together to create a qualia in the big, broad sense; information, which information is available to you; integration of your experience in your whole situation; and exclusion, sort of what conditions are impossible to attain. And you take these things into account when you're making these mix. And then these guys conjectured that the mix should form a category and describe sort of what the morphisms might be and composition, and that the qualia should as well. And there they said, well, the morphisms, we don't know. And maybe there's a functor between this more convincing category and the less convincing maybe category. And that should be, these people they're neutral on the notion of whether this IIT is actually correct. And they say one way of testing whether it actually is correct would be to say, well, is there some functorial way, some way representing, respecting composition to relate? So this is a recent article from 2016. Maybe there's something that could be pursued in that direction for those who are more philosophically and cognitively minded.
All of these models seem to be published in neuroscience journals rather than category journals, which doesn't reassure me as to the category theory.
Well, so the previous one, actually, so each one, of course, has to explain the category theory. And the previous one actually explained it in a competent manner. So I think that, yeah. No, I agree. I think.
It's not going to show up in categories, it's not research on categories.
Exactly.
It's research on cognition.
Yeah.
It's application of categories.
Yeah.
They have to explain the category.
Yeah, they have to explain the category theory.
They often
Yeah.
what we think of as an undue amount of time doing that, but otherwise, no one
Yeah. Exactly. So that's another direction in which people have looked. And then there's an entire book called A New Foundation for Representation in Cognitive and Brain Science. So this guy, Javier Gomez Ramirez, he's actually worked with Arisman. And he had adopted a lot of ideas from her memory evaluative neural systems into thinking about, so his PhD was actually in neuroscience. And he wrote, this book is essentially his PhD thesis, I believe. And so what he says is, okay, well, let's look at the directed graph of a network of neurons. Okay, so we're taking sort of this sort of perspective on what a neuron is. And putting them together, you get a directed graph, and from that you can make a category, obviously. And so let's look at that. And he says something about kind of enumerating the co-limits or limits in this category. I'm not exactly, I would have to read the whole book. But basically, he's interested in what sort of limits and co-limits you can create in this category.
And his idea is that you should be thinking about the hippocampus. This is, again, from the visualization team at Blue Brain. They did a very nice model of a hippocampus. The hippocampus is part of your brain that's involved with both localization in space and memory. You know, this is one of these reasons why if we want to remember something, it's often useful to link a memory to a place or to a thing, because you're actually using the same part of the brain for that. But in any case, within this hippocampus, there are what are called place fields, place neurons, that will fire when you're in a specific place. And his idea is that these sort of place fields, they should be built up what are called grid cells. So there were people, the 2014, I think, Nobel Prize in Medicine was given to neuroscientists who were working on these grid cells, which actually give you, they fire in response to a certain hexagonal grid in space. And his idea is that somehow you should be able to take colimits of the network of grid cells in order to build up place fields. And I think it's probably worth exploring further. Maybe there's some connection. I mean, the point is that there are actually place cells that fire when you're in a certain place. But maybe another way to detect a place is to think about colimit of grid cells. I don't know. But it's an interesting hypothesis, and it's actually something that you could perhaps test. And so... And he seems very, very enthusiastic about working with category theorists. He actually reached out to me at one point, and I was really too busy to discuss with him. So if anybody's interested, I mean, he'd be delighted if a category theorist would reach out to him and want to work with him. So, and finally, I want to talk about work of Sophie Rayner, who was here for the workshop last week and most of this week. So she did her PhD thesis with me and with Ron Levy. And at the beginning, it was meant to be a thesis about kind of category theory in neuroscience, and it became a thesis about category theory. Because we were thinking about, well, how can we model hierarchical networks, and how can we model learning in hierarchical networks? And in the end, it became a thesis about closed multi-categories. But you know, the basic idea is you're starting with a piece like this, where you have this sort of, you're thinking about a network that has a natural hierarchy to it, and you want to understand mathematically what is the right framework in which to be thinking about these hierarchical structures. And you know, how could you do something like modeling learning or dynamics in such a network? And so you really want to understand what is the correct mathematical framework. And so we're talking about these modular networks, and I said to her, I should maybe never have said this, I said, what is the connection between modular networks and modular operads, besides the fact that you're using the same adjective? Turns out there is a connection. And that led to her entire 250-page thesis or something like this. So we're starting with something like this. And there are various ways you can be thinking about this kind of structure. One is in this sort of colimit kind of point of view. You could say, well, I'm starting off with a bunch of elementary pieces. I could think of those as my neurons with their axons and dendrites. And then they're being connected together to form little units, processing units. And then these things are being put together as, so here, the idea here is that I'm saying, well, I'm going to forget the internal structure here and just remember that I have 3 edges that are outgoing. And I'll forget the internal structure here, remember that I have 4 edges that are outgoing, and then I have a way of gluing these together and it forms a bigger unit, and so on. And so the idea is you can think about this as being, again, built by these colimits of colimits of colimits kind of construction. That's one way to think about these things. Another way is to say, let's zoom out. You know, if I go back to this thing, I've got sort of one blob that has 3 outgoing edges. And I can instead say, well, I'll look at this thing with the 3 outgoing edges, and then I zoom in, and I zoom in, and I zoom in. So you have sort of this idea of having increasingly complex, increasingly fine vision of what it is that's going on. Or you can say, well, instead I can think about sort of a collapsing process instead. Say, I'm not going to build it up as a colimit. I'm going to think I start with the whole thing. And then I just say, I'm going to start with this graph. This is my original graph. And it's some sort of, the next hierarchical level, I'm going to take these things and collapse them out. And so then I just think about these things as vertices. And at the next stage, I'll collapse this out. And I think of these things as vertices, and so on. So you have this way of making a hierarchy of graphs, where at each stage, you've just collapsed the modules at the previous stage. Try to understand this sort of hierarchy of graphs in this way. So that was what she studied, and a couple of the results that one can extract from the very abstract mathematics there is that if you have, so there is a monad that's describing 3 modular operads. And it acts on the category of graphical species. So these are some pre-sheaves on some category of elementary graphs. So these are corollas and the free edge. So if you take a graphical species, a pre-sheaf on this category of elementary graphs, and you iterate this free modular operad monad, then an element of this thing is really one of these k-level hierarchical graphs. This is this last stage where you're collapsing and collapsing and collapsing. So that gives us a nice mathematical framework in which to understand these hierarchical graphs. And one connection that she observed is that there's an operad that governs uncolored modular operads, and it turns out there's an operad, a sub-operad of David's operad of wiring diagrams. So there's a connection between, and this also shouldn't be surprising, there should be a connection between wiring diagrams and this sort of situation. So this is a kind of very formal categorical framework is already available to us. Aha. Let's speculate. So what I'm going to present in the next couple of slides is really just some ideas I'm throwing out there about what might be potential frameworks, and then what are some of the important things you need to keep in mind if you want to try to apply these frameworks. So just as a reminder, the big question for the neuroscientists is really this. How does the structure of the connectome, and here I can mean the connectome on the level of neurons or the connectome on the level of brain regions, how does it shape its function? Because it's one thing to say, okay, I have a beautiful picture of the architecture of the brain, now but I'd really like to understand how it works. That's the point, after all. And then with plasticity, with learning, we see that the structure changes. So what I was talking about earlier was the fact that these synapses can become stronger and weaker. Sometimes they can even disappear completely. So you can have a connection between two neurons that was there and is there no longer in result of plasticity. So you can think, how does it model their structure? Those are sort of the big questions that the neuroscientists are interested in. Possible categorical frameworks, there are lots. If you think about vibrations, you can think about, you could have some version of this, of categories in which you could say, I have the underlying structural situation, and then I can decorate it in various ways. I can say I'm going to decorate it with various vector spaces or whatever. And think about vibrations. I can think about props. Props come up very naturally. If you're thinking about these neurons, they have all these inputs and outputs. You have a bunch of props that you're gluing together. Maybe there's a way to think about that. Because time is an extremely important element in brain function. It really is all about what's going on through time. Maybe there's some of David's ideas about using topoi that capture temporal elements that might be interesting. One point, Ron Levy and I were thinking about the idea of having sort of field theories on the brain, sort of a la spin networks and spin foams and things like that. That might be a way to understand what's going on with plasticity. I don't know how completely justified that is, because I don't know whether it makes sense to require that, even if you can think about the brain as being put together by doing some sort of concatenation or some sort of both a tensor product with putting two things next to each other, concatenation and gluing things together and so on. You have this way of understanding a network of neurons as being built up by something like that. Whether when you're looking at a sort of field theory on the brain, you really want to have a function that's necessarily going to be monoidal or something like that, I don't know. Higher categories should come into this in some way, because it's really, I think that when you start putting networks of neurons together, it's not simply a question of looking at whether you have paths to get from here to there, but the relations between the paths and sort of higher cell structures, I think, should be important, and particularly maybe infinity categories, because when we're thinking about directed things, we're thinking about a directed network like this. If you want to if you think about quasi-categories, they give you a way of actually capturing this kind of directed structure, perhaps. There are also the compositional networks of Brandon and John that might be interesting for thinking about something like this. And then listening to Dan's talk yesterday, I thought, well, he's got these ways of working with digital circuits. Maybe there are some ways to create an analog version for the brain, which would also have to take into account the stochasticity. But I think there are lots of possible categorical frameworks that could be interesting here. I haven't spent very much time at all thinking about how to do so, since I've been focused on thinking about how to apply topology in this context, but I think there's plenty of possibilities. Important issues to keep in mind when you do so is the fact that the structure of the brain is so multi-scale. Everything from the level of neurons which are very, very small, to the level of entire brain regions? Do you want to think about an entire hierarchical structure? At what level do you want to focus on it? Do you want to think about a neuron and a connection between two neurons like this? Or do you want to take into account all the complexity of the dendritic arborization? Do you want to take into account the weight? So there are various levels on which one can be working here. And you can learn things from focusing on any particular of them or from building in a lot of them. Stochasticity is essential. If the brain were completely deterministic, it wouldn't be nearly as powerful as it is. So that if you really want to do a good model of a brain, there's got to be some way to build stochasticity into it. Maybe some sort of probability methods. In discussions with Jamie 2 days ago, we were talking about the redundancy that comes up in biology. If you had just one neuron that was responsible for remembering how to integrate 1/x, you'd have to be really careful about not drinking too much alcohol. So your brain has a lot of redundancy built into it because this provides you with robustness. And your brain hasn't always figured out, evolution hasn't always driven your brain to doing things in the most efficient way, the most beautiful way, the most whatever. The evolution has driven your brain to be able to function, to do what it has to do. So what you think of as the most elegant solution is probably not what Mother Nature has come up with. So, and Mother Nature's figured out 100 different ways to do it and has built 50 of them into your brain, something like this. So that's, if you want to be really biological, that's something. Plasticity. How can we, what is the right categorical framework in which to describe what's going on with a network in which weights are becoming smaller and smaller and maybe even disappearing, or new connections are forming? This is something that would be really beautiful to model. Dynamics. If we want to talk about activity, then we're talking about dynamics in the network. And you have to take into account the fact that, at least when you're trying to do things from a sort of topological point of view, it's not always easy to figure out what dynamics. So I just want to give a simple example of something that happens with 4 neurons. The sort of problem that comes up with dynamics. So we can make directed graphs linking these guys in various ways. So you can have reciprocal connections between neurons, that's no problem. And we can do something like this. And again, a reciprocal connection between these two, and then something like this. So there's a way of associating to any directed graph like this a simplicial complex using directed click complexes. And in each case, if you take the geometric realization, it's just going to give you a 2-sphere in this case. You have 4 triangles that are being glued together to make a 2-sphere. However, if you look at what's going on dynamically in these 3 pictures, it's very different. Here we have everything that's sort of flowing down here to a sort of fixed point. Maybe things will go around this loop for a little while. Here you have 2 fixed points with this periodic behavior in the middle, and here everything is converging to the middle. So we have 3 very different dynamical behaviors that need to be taken into account, that need to be captured somehow. And this is one thing we're thinking about on the topological side as well. We have a bunch of tools that will allow us to distinguish between this one and these 2, but distinguishing between these 2 turns out to be remarkably difficult using the tools. So we need to find a nice way to capture dynamics. As I said earlier, temporal structure is extremely important. And synchronization of electrical activity in the brain also plays a really important role in brain function. So, I'm reminded of, to say the word synchronization by Dan's talk yesterday. This is something that's an essential element in brain function. So, I think I am actually out of time, so topology won't reveal anything. Unless... Okay, what topology reveals? Let me tell you a little bit of the story. So I'm going to say a little bit more about what it is that the Blue Brain Project has actually done. So they've made this digital reconstruction of part of the somatosensory cortex of a 14-year-old rat. The somatosensory cortex would be roughly here in my brain. If I were, it is. I have one too. In the rat's brain, it takes in incoming sensory input from the thalamus. So the thalamus is sort of your relay between the outside world and the brain, and the thalamus sends information. The somatosensory cortex starts processing it, saying, what did I just touch? Was it hot? Was it cold? Was it sharp? Things like that. And then they built it so that it can you can simulate both spontaneous, random firing of the neurons, and evoked activity. So here we see the different layers. This is the top layer, the outside, layer 1, down to layer 6. And so they took into account a whole lot of biological detail, making this reconstruction, including precise neuron morphologies and really explicit connection probabilities. And so they took parameters for this algorithm from 5 different rats who were sacrificed at 14 days of age, and measured all these different parameters. And then using these parameters, and the algorithm, the very complex algorithm they had, they made 7 different instantiations of this bit of cortical tissue for each of these 5 rats, and then took the average values as well and made it substantiation. They ended up with 42 examples of these microcircuits, each of which is about 31,000 neurons, and about 8 million connections. And I remind you that each connection is actually made up of several synapses, so it corresponds to roughly 37 million synapses. And then there is a certain amount of biological data that's built into the microcircuit, and then it was validated on biological data which hadn't been built into the circuit, and they did a very good job of reproducing this data. And if you want to look at the adjacency matrices and play with them, or look at more information, you can look on this portal that they created, where you can actually find out all sorts of things about what the neurons are, how they behave, and so on, and how they're all connected for all 42 networks. So what you do is you associate two, I said we're going to look at this as a directed graph, we associate two directed complex. So if we have a directed graph with vertices, edges, and this function is telling us the source and target vertices, we can make a simplicial complex, the 0 simplices of which are just the vertices, and n simplices of which are sequences of vertices, sequences of n plus 1 vertices, such that every time that I is less than j, vij is an edge. So we're taking things into account. So if we're looking on the level of 2 simplices, so if I have 3 neurons, something like that is going to give me a 2 simplex because it's a totally ordered set of neurons. Whereas if I look at this, it's not going to give me a 2 simplex because it's a cycle. So we build these in. And so these are precisely the acyclic cliques of the graph, or another way to say it is the click so that every subclick has one source and one sink. And so this is examples of how you go from a directed graph to one of these simplicial complexes. So if we look at this one, for example, one of these triangles, this one here, this is a cycle. So this is not going to give us 2 simplex, whereas this one, you actually have a source and a sink. And so this triangle here, like this, is going to give us a 2 simplex. So this is a way we have of associating a simplicial complex to a directed graph. Taking into account, if you think about these n plus 1 directed clicks as sort of being higher dimensional versions of directed edges. And then we quantified the structural complexity of these networks. So we did a comparison of the actual blue-brain microcircuit with various null models. And so the blue-brain microcircuit is here in blue. And what they count is, in terms of simplex dimension, the numbers of simplices. And what we see is that we have roughly 75 million 2 simplices, roughly 65 million 3 simplices, and so on. And we even have some 7 simplices and plenty of 6 simplices. Whereas the null models would be, for example, an Irish-Renyi graph, where you just take 31,000 vertices with a connection probability equal to the average connection probability in the microcircuit, then it dies out much faster, it dies out after 3. And these two null models here, the ones related to not taking the very specific morphologies of the neurons into account, sort of fuzzing things out a little bit, and it gets much less complex. And it turns out these simplices actually have some sort of functional role as well. This is about the correlation in the electrical activity between 2 neurons that have some connection. And what you see is that what this dimension here is the dimension of the maximal, it's the maximal dimension of a simplex to which an edge belongs. And the higher that dimension is, the more highly correlated the electrical behavior of the neurons is. On the other hand, go down to this end. What is that point there saying? It's saying, I have a connection between 2 neurons that doesn't belong to anything bigger. It's just by itself. It's just a little hair in the middle of the network. That its correlation is lower than, well, this dotted line here is the average correlation in spiking behavior between 2 connected neurons. And if it means that you're connected, but you're not part of something bigger, your correlation is actually going to be lower than average correlation. So you need to be part of something bigger in order to. So that's an indication of a functional role for simplices. And you can actually do using things like calculating the Betty numbers of these complexes. And we did that by rat. So we had these 5 different rats and the average rat, and plotted Betty 2 versus Betty 3. And it basically is very beautiful clusters. So somehow the topological invariants are detecting the biological differences between these different circuits. So topology is picking up something biological.
Wait, so each of those rats has about 7 blobs associated.
Exactly.
What are those 7 blobs?
So there are 7 different instantiations of the circuit for the parameters. So I take the parameters from an individual rat, make 7 different instantiations using those parameters because the algorithm itself, construction is stochastic. Okay. So it's interesting to see, first of all, that they do cluster closely together and that they are separated. So it's somehow this is nice biological medium. Let's see. Oh, you also get something like this for, this is showing activity in the circuit. And so this is just a bunch of these together, and you see sort of this wave of activity. And what you want to do is sort of quantify how this activity is working. And I don't want to abuse too much. Let's see. And so you can encode that sort of activity by making a time series of directed graphs by looking at in each time bins. In each time bin, you look at the subgraph of sort of active connections in the graph according to a certain transmission response rule. So you start with the structural graph, and you're at each stage you're having some sort of subgraph which is describing which part of the graph is active. And then that gives you a time series. Since you have a time series of subgraphs, you have a time series of simplicial complexes, and you can look at what's going on with the numbers of 1 simplices, Betty 1, Betty 3, Euler characteristic, whatever you want. And this was for 9 different input stimuli. And I think I'll conclude here with the swoosh. So what we have here is we have 3 different stimuli that we're looking at, and we're looking at Betty 1 versus Betty 3. Why not? And you see you have this sort of, as time passes, so you have time moving along in this direction. As time passes, you have increasingly complex structure. You have the Betty 1 that's increasing, and then just as Betty 1 starts decreasing, Betty 3 starts increasing. So you have this you have a bunch of loops that appear, and then you have a bunch of, we could put Betty 2 in there as well, which would be the 2-dimensional cavities, and 3-dimensional cavities that appear. And you have this whole process of increasingly complex structure that at some point just sort of collapses when you've sort of finished processing it. So that's the sort of thing that we can detect with somehow some sort of quantification of what's going on during information processing during activity. So thank you very much. This is where I work when I'm being a neuroscientist at the Blue Brain headquarters at Campus Biotech, and thank you for your attention.